Thanks so much for the invitation. It's been so nice to be here so far. Had a great time. I've really enjoyed the talks. Today I'm going to be talking to you about maternal transmission, sex ratio distortion, and mitochondria. And I've been working for a long time now on facultative inherited symbionts of insects. Here are a bunch of host insects that we've been working on. So symbionts that are similar to Wolbachia that you can see here in a wasp egg that are transmitted maternally, so primarily in the egg cytoplasm. Today I'm going to be talking more about mitochondria than about inherited symbionts. And so first, I'll just start by trying to make some parallels between inherited symbionts and organelles with a focus on multicellular and also mitochondria over chloroplasts. So focusing on sex-limited transmission and primarily female transmission. So I'll talk about briefly a few patterns and causes, and then three potential consequences of female-limited transmission. And then I'll end with a case study of sex ratio distortion in a book louse, a tiny little insect. So we've known for a long time, and lots of folks have thought about this, that uniparental transmission of organelles and symbionts is very common. And so why is that? So our first intuition might be that it's about size, and that's certainly not the case, that there's less room in sperm, so that less things are transmitted, or that they might be swamped out by the egg. And that's certainly not the case. Lots of exceptions, and useful to think about gymnosperms, for example, and plants in general, where we see interesting species-specific patterns where you have either mitochondria and or chloroplasts that are transmitted either exclusively through the male and or the female. So it's not just about size. Indeed, it turns out that there's that the control over transmission is very specific and very selective. And so here you can see a nice paper from Sarah Al-Rawi looking at C. elegans. And so what you can see here is just after fertilization, to note that the autophagy machinery is turned on and active and is specifically recognizing and destroying mitochondria that are associated with sperm, mitochondria that are associated with the male. So there's very, very specific control and recognition. So a number of ideas about what's driving that. And one important pattern and idea is that uniparental transmission of mitochondria reduces heteroplasmy and potential competition. We know even without uniparental transmission that heteroplasmy is common, is pervasive, and with important consequences. So even without control, we know that mitochondrial variants with potentially negative effects on fitness and sometimes a replication advantage can increase in an individual. So here, for example, is just an image of a human skeletal muscle that is polymorphic, and it's stained for cytochrome oxidase activity. And so you can see that the blue cells are not active, and that relates to those cells containing heteroplasmy, a mitochondrial variant that has a mutation in a tRNA. So here's one interesting exception, an exception that maybe proves the rule, and that's a common pattern in freshwater clams and their relatives called doubly uniparental inheritance. And so in freshwater clams, mussels, for example, there are two kinds of mitochondria, so a maternal and a paternal type. So here you can see, for example, a southern from Zuros, and the two mitochondrial types are really interesting. So the one type in the females is transmitted transmitted from mothers both to sons and daughters, whereas the male type is transmitted, the male type here is transmitted from dads to their sons. And this has been associated with a lot of interesting outcomes. So the two different mitochondrial types don't see each other, they're highly divergent, and they contain novel genes. So here you can see from Sophie Breton's paper, for example, that the male-specific mitochondrial genome, animal mitochondrial genomes, tend to be called boring, but in this case, it's acquired some novel genes. So here, male-specific, and the same thing has happened with the female-specific, female-limited mitochondria. And so it's been thought that the male type has basically avoided recognition and degradation in terms of host control. So what about symbionts? Certainly much less is known in terms of control, in terms of symbiont transmission. Lots and lots of cases, of course, of almost exclusive maternal transmission. But there are also some interesting mixed systems which I think are really promising for future study. So for instance, here's one. So Regiella, for instance, is a maternally transmitted facultative symbiont in aphids, and it's got both maternal transmission, so here you can see it via embryos, and it's also, interestingly, transmitted sexually, so paternal transmission. Here you can see it aggregated in male accessory glands. So I think that these systems are going to be very useful to look at interactions between the two different forms of transmission. So that's all I'll say about causes of uniparental transmission. I just thought I'd also put up some unresolved or questions that may be useful. So certainly symbionts, much less has been worked on relative to mitochondria. So how pervasive is symbiont heteroplasmy? We see inspired by John McCutcheon's talk. And how does it affect host symbiont host fitness and symbiont replication? And then do hosts use the same machinery to control mitochondria and symbionts? There have been a couple of recent interesting papers showing how the autophagy pathway seems to be important in controlling symbionts, and so there are potential interesting parallels there. So I'll briefly talk about a few interesting consequences of maternal transmission. And one is increased transmission via females. So well known, well studied, this idea that symbionts that are almost exclusively transmitted through females, males are an evolutionary dead end, and so they increase transmission by distorting and manipulating reproduction. So very well characterized, of course, in inherited symbionts and insects. It's evolved independently many, many times, bacteria and microsporidian symbionts that do this by killing males or by transforming potentially male hosts into females that then can transmit the symbiont. So for instance, here a wasp that is infected with an inherited rickettsia that causes parthenogenesis. What about organelles? Well, certainly we see the same patterns, and the best examples are cytoplasmic male sterility, where you have repeated evolution in plants of mitochondrial haplotypes that in all sorts of different ways either cause inviability of pollen or affect anther development. So it's evolved independently numerous times, and interestingly, it's been accompanied by repeated evolution of nuclear suppressor genes, so races between and cycles between mitochondrial and nuclear genes. And another interesting parallel is that it's involved also novel mitochondrial genes. So for instance, a characterized case of cytoplasmic male sterility in rice, where It's caused by a novel chimeric gene, this WA352. And here you can see that it causes infiable, non-staining pollen compared to the controls. So that's all I'll say in terms of increased transmission. In general, a big question in the field is how pervasive are cytoplasmic sex disorders, and have they shaped the evolution of sex determination systems? We see how sex determination systems evolve very rapidly, and it's been speculated repeatedly in the literature that this is driven by cytoplasmic sex disorders, in the same idea of these races between nuclear suppressors. It's also, for example, been speculated that these male-specific mitochondria in the freshwater clams represent a novel sex determination system. Another question is, why don't we see mitochondrial sex disorders in animals? And there's certainly been speculation about that. The next consequence that I won't say too much about, we've certainly seen yesterday how maternal transmission is associated with increases in deleterious mutations. Here we also see accumulation of male-specific deleterious mutations. So again, because transmission is through females, we can imagine that variants that might reduce fitness in males but don't reduce fitness in females might increase in frequency. And certainly lots of examples of mitochondrial mutations that cause abnormal problems in human male fertility, for example. So in general, less work has been done on this question with inherited symbionts, and so that's sort of also, I think, a useful and open question is how much evidence do we see for inherited symbionts showing reduced function in males? The last consequence I'll talk about is co-transmission. So again, this is something that has been certainly appreciated, is that because the inherited symbionts and mitochondria are co-transmitted, their fates are intimately linked. And one example is one that we've worked on with John Janicki at the University of Rochester, where we've characterized an inherited symbiosis, a spiroplasma symbiont. So here you can see it here, a symbiont that protects its host, a mushroom-feeding Drosophila, from a virulent parasitic nematode. And what we've found is that that symbiont is fairly recent in terms of its effect. It's conferring such a strong benefit that it's spreading across North America. So you can see here, quite common in eastern North America, less so in central, and until recently there were no spiroplasma-infected flies west of the Rockies. And so as it's spreading, it's also bringing its mitochondrial haplotype along with it. There's a whole diversity of mitochondrial haplotypes west of the Rockies that we predict are going to go extinct. So some unresolved questions are, does the fate of an inherited symbiont depend on the mitochondrial genome, the mitochondrial haplotype that it's associated with, and vice versa? And also, is there co-adaptation between specific symbiont and mitochondrial haplotypes? So we've been thinking about these different pieces of the puzzle, and I wanted to tell you a recent story about extreme mitochondrial polymorphism associated with a sex ratio distortion in an insect. And this work has been done by 2 graduate students in the lab, Christina Hodson and Finn Hamilton. So the story starts with a book louse, so Lipocilus bostricophila. And so this is in the lineage of insects that contains book lice, bark lice, and parasitic lice. And Lipocilus bostricophila is a cosmopolitan pest of stored grains found throughout the world, found in 1/3 of kitchens in England, found in my kitchen, and here you can see it right here. Interestingly, it's parthenogenetic, it's 100% female, and 100% infected with a strain of rickettsia whose role is a complete mystery. And there's speculation that the rickettsia is obligate, there's speculation that it's associated with the parthenogenesis, but no support for either of those. So we were very excited to find the sexual sister of Lipocilus bostricophila. This was initially found by Adi Behar, a postdoc in my lab, and then Ed Mockford, who is the guru, the god of SOCID taxonomy and systematics. And it turns out that this book louse, we found it in one of my favorite places in the world, in the Chiricahua Mountains of southeastern Arizona, and it really likes to hang out under dead yucca leaves. And here's just to convince you that it is indeed the sister of the asexual cosmopolitan Lipocilus bostricophila. So we were really interested in learning more about it and studying it in more detail. So we brought it into the lab, and we found that it has a very unusual reproductive biology. So these experiments were done with Christina and Ed, and then some initial experiments were with a collaborator at Oklahoma State, at George Opit. And so here you can see it here in our lab cultures, hanging out on Rice Krispies. You can see those tiny little book lice. And so what we found is that it's an obligate sexual. We confirmed that. It's not infected with rickettsia. And then we found that our lab cultures were polymorphic for this extreme sex ratio distortion. So basically, there are two types of females in our cultures. There are ones that produce males. There is a female bias, but they do consistently produce males. And then there are ones that never, ever produce females. And this trait is strictly maternal, so that females from this line will only produce daughters if they've got males from this line to mate with, and we've maintained this in the lab separately for a long time. So also to confirm the exclusive maternal transmission, we found that it's associated with highly divergent mitochondrial haplotypes. So this was some initial CO1 sequence. So you could see here's the asexual again, and here's CO1 sequence from the two different types of females in the population. So one thing that we wanted to make sure was that this wasn't a case of sperm parasitism. There's some, the best studied, for example, are fish systems where you have something that's essentially parthenogenetic, but it needs fertilization from a close relative to trigger development. So we ruled that out. So confirmed that there's gene flow between the female-only individuals and the mixed individuals. So here you can see two tightly linked SNPs, and across from a female-only line, a mother with a father, that the daughter is clearly incorporating alleles from the dad. So we're really interested in this extreme maternal sex ratio distortion. We want to understand the ecology, how on earth does it persist, and we also want to try to understand the genetic basis of this distortion. So, of course, it screams microbe, especially from where I'm sitting, and female maternally transmitted sex ratio distortion. And we've tried lots and lots of different ways. We've done microscopy, and then we've done targeted and non-targeted searches. We've also done some Illumina sequencing, and we haven't found any evidence of a microbe. So here, for example, are just some bacterial profiles of 16S RNA comparing the two different types of females. We don't see any signal of any microbe in this system. So probably not, but that leaves us with two possibilities. One is that it might be a selfish genetic element. So our Illumina sequence We don't see signal of a microbe. We also don't see signal that that the nuclear genomes of the two different types of females are very different. So it doesn't suggest that the distorting female, for instance, is a hybridogen or sort of a hybrid genotype with an odd distortion. But we do see a few sequences, very few, that seem to be unique to the distorter and that are not found in the normal female, and not vice versa. So we're going to do some cytology and some deeper sequencing to follow up on that. So that leaves the mitochondria, and we were really interested by these deeply divergent mitochondrial CO1 sequences. So we spent quite a bit of time trying to sequence the mitochondrial genomes of these two different types of females. And we thought it would be easier than it was, especially since animal mitochondria and arthropod mitochondria are quite boring, as David said yesterday. But it turns out that sosids have quite unusual mitochondrial genomes. And we've seen before, so this is mostly Renfu Xiao who's done this work. And here's an image from one of his papers. So most arthropod genomes, most animal genomes are these single circles. And you can see this variation in this lineage. So Lipocellus, so the asexual, its mitochondrial genome has been sequenced, and it consists of 2 circles, and all the way up to human body lice, where their mitochondrial genomes are these mini circles, 20. So in combination of Illumina and Sanger sequencing, we spent a lot of time trying to close and get a sense of the number of circles. And so here's just to show, again, the mitochondrial divergence between the two different lineages and compared to the lack of divergence in the nuclear part of the genome. So comparing, for instance, the normal versus the distorter and back relative to comparisons of mitochondrial sequences. So we found very, very high mitochondrial polymorphism between the two different types of females. So here you can see these female-only individuals and then these mixed quote-unquote normal individuals. So they differ in the number of circles, and they differ in the order of genes, and we haven't completely finished closing, but we're pretty close. So in parallel, we've also found one other interesting polymorphism related to the mitochondria. And this started from, so it was with Brent Gowan, who is our EM specialist in the department. And this started with our searches for microbes. We didn't find any microbes, but we found one interesting consistent mitochondrial morphological difference. So in age-matched individuals across all tissues, the mitochondria look the same. They look quote-unquote normal, except for one place. And that place is this structure here, which in the literature has been called a rectal gland. It's packed with mitochondria, metabolically active. And so here you can see in age-matched, so 4-week-old individuals, the mixed male-producing book lice have got packed with mitochondria, Christie, happy-looking mitochondria, whereas the female-only individuals completely different. So we might hypothesize that there's impaired function in these distorting females, which might explain the ecology of how these are balanced. And interestingly, the female-only, so females from the distorter line, die much sooner. We found other differences. We haven't found differences in fecundity, taking away the survival differences. There's a difference in survival time. But what you can see, the striking difference, amazing how long they live all together. A lot of the normal females are still alive, But after about between 100 and 150 days, we see this vast decline in the distorting females. So lots to do, but our working hypothesis is that the female-only sex ratio distortion is caused by a maternally transmitted selfish genetic element, and that the distortion persists as a polymorphism due to negative fitness consequences of the mitochondrial variant that's associated with it. And so those are the directions that we're headed to try to solve that. So I'll end there. Hopefully I've convinced you of a few more important parallels between mitochondria and symbionts driven by maternal transmission, and certainly lots of cryptic diversity, maybe within species diversity, of course, that's waiting to be uncovered. Also waiting to see when we'll see more mitochondrial sex disorders, maybe ones that have acquired a novel gene, possibly from a symbiont. And again, lots of folks to thank, and you too.